Great. Good morning, everybody, or good afternoon, depending from where you are assisting today's presentation. Today we're having our guest, our speaker for today. It's Daniela Vargas. She's gonna present the work she did during her postdoc at the Universidad Autónoma Metropolitana de México. So the second week that we have a presentation from Mexico, although Daniela is a biologist from Venezuela. Actually, we were colleagues. We studied together at the Universidad de los Andes, where she got her degree in biology. There, if I remember well, she worked with anti-malaria drug resistance, and then went on to Amazonas, Venezuelan Amazonas, to continue her work, and then where she developed her PhD, although she coursed her PhD at the Universidad of Puerto Rico. And then after finishing her PhD, she went to Mexico, where she did the work she's presenting to us today. So for me, it's a great pleasure to introduce and present a colleague of mine to talk at the EcoEncontros. So without further ado, thanks again, Daniela, for taking the time to present this work, which is just recently came out.
Thank you, Adriana, and thank you everybody from the University of Sao Paulo. Thank you for the invitation. So what I'm going to present you is a study that we perform in the subway of the Mexico cities, and we wanted to know what was the microbiome over there and what was the interaction among this microbiome and the passengers. So as Adrian said, this was a postdoctoral research of 1.5 years, and this was performed by the Universidad Autónoma Metropolitana 1, and with the guidance of Dr. Mariana Painberg, that she was the director of the study, and in the team were Carolina González, Apolina Hernández, and Luis Alcaraz, and this last one from the la Universidad Nacional Autónoma de México. And so we already published one article of this study, and this one that I'm going to present to you, it's already a preprint in PLOS ONE journal. So as an introduction, we have that in the last 7 years, subway microbiota studies have been performed in some cities. All of them have used bacterial sequencing approaches instead of culture-based, since it better recovers the microbial diversity. So Mexico City's study is the first one, including in surface and human interaction. And these types of studies helps to better understand the relationships that the microbes have with our environment and themselves, with the people. So in particular, the subway has several characteristics that have microbiological implications and that make it a unique environment. So it is a public place with no sunlight, and this has a very important effect because the sunlight, remember that it is a natural sterilizer, and it has also a high affluence of people, particularly in the Mexico City subway is the 9th most visited subway in the world. So it brings around 5 million passengers daily. So also that quantity of that many visitors promote the interaction of people, the contact among people. Also, this subway brings into the city people from multiple areas around the city, including semi-rural areas. And the exterior air is brought into the subway from ducted airstreams. Also, the food and beverage are sold inside the stations and even inside the trains by normal vendors. The cleaning, it's very variable, and it can be from the use of chlorine to just water. It varies a lot in between stations, and it is performed with a non-strict schedule. And the cleaning, and the woman, and differently to other subways in the world, and this subway has the three first wagons or cars in the train are exclusively for the use of women and disabled people. So all these characteristics makes this public system a very complex and interesting place to study interactions. And as we know already from different studies, the microbiome or the microbiota of the interior of a building is formed by the microbiota of their occupants. So this makes possible to detect signs of cohabitations. And as you can see in the picture where you have a person that is almost green, all the green color that you see is the bacteria. It reflects the bacteria that they have inside, and all the spots that you see around the room are the bacteria that are sprayed out by the humans that we usually sprayed out in the areas that we have, that we inhabit. That's why you can match a person to the house where that person lives, because they are more similar, more than anyone else that doesn't live in that house. So we first want to know what was the microbiome. Yeah.
Sorry, one question. Are you changing this? Okay, this slides was okay.
Yes, I think because of the connection. Yeah.
Okay.
So you can see it. Okay, thank you. So yeah, we wanted to see first what was the composition of the microbiome in the subway of the Mexico cities. So what we first did was to sample turnstiles that are similar to those that you can see in the picture, and also poles. The poles, or the in the plot you see it as handrails, so it will be the hundreds of poles inside the trains. So we took samples, around 23 samples from these two surfaces, and what we saw is that the surfaces were dominated by human, that would be skin and saliva bacteria, but also by soil bacteria. So we also saw that it has a very different composition, handrails or poles, and had a very low, not very low, but a lower diversity than the turnstiles. And also both had a very different compositions. So one of the hypotheses is that two factors are playing an important role there, that it will be the type of use that the people are doing with the surfaces, and also the material of the surfaces and the type of use. I mean by that you can touch a surface with the hands, but also you can touch it only with the cloth. Okay, so depending on how you touch it, you will find that a particular microbiome in a particular area. So what we saw is that people from that the turnstiles were mostly touched by hands, but also by cloth, while the poles were mostly touched by hands, only by hands. So we wanted to go further. So we asked these questions that were that we try to answer in this next paper, this next study. So we wanted to know what is the composition of other commonly touched surfaces. What is the effect of the surface cleaning, and how fast the microbiota can be recovered? Which are the interactions among surfaces and passengers? Can we detect cohabitation sign in woman-only wagons in comparison with a regular wagon? And what are the effects of traveling on the passenger's microbiota? So for answering this, we designed this study that I think that you will see it very soon. Okay, so we have that in the first panel. You can see different spots, red spots, and those were the areas or the surface that we sampled. We took around 5 to 8 samples per surface. So we first sampled turnstiles, then escalator handrails and stairs handrails, and a platform floor. We also sampled seats and poles, and we did the comparison, as I told you, from the women-only wagons with the regular wagons, waiting to find vaginal indicators, a bacteria that can be matched with a microbiota typical from women. Then in the panel B, we sampled, we took a sample from poles, and we then cleaned that surface, and we took sample in several time points after cleaning. We performed this with a sequencing-based analysis, but also we did it with culturing. That would be the blue spots. And in the panel C, we took samples from 8 volunteers and samples from the hand, and then we took samples after they traveled. And we performed the same experiment, but with a hand washing procedure before traveling. So these are the methods. We performed the swabbing of those surfaces in around 100 square centimeters of the surfaces with nylon swabs. We recorded temperature and humidity in each sampling site. We took a total of 50 samples, and for culture-based analysis, and we grew those bacteria in nutrient agar. We took a total of 89 samples for DNA sequencing, and we performed the DNA extraction with power soil kit. We performed PCR, so we did the amplification of V3B4 16S ribosomal RNA gene. We then did the sequencing with Illumina platform, and we performed the bioinformatics with, we performed the ratification, and we assigned taxonomy with green genes, and the analysis were performed with operational taxonomic units and ASV, that these two are operational definition of bacteria that we usually use it when you are analyzing bacteria based on sequence-based analysis. And then finally, we processed this data, and all the statistical analysis was performed in R. So here we have the results. You can see here taxonomy summary. where each of the bars correspond to each of the different surfaces that we sampled. And we can see that a big proportion, a high proportion of bacteria correspond to taxa that are present in very low abundance. That will be the y-axis. So, and here we have in asterisks those bacteria that are in the top 5 with the highest relative abundance. That will be Acinetobacter, Corynebacterium, Propionibacterium, Staphylococcus, Streptococcus, that are basically bacteria typically found in human and environmental bacteria. Here you have a plot of beta and alpha microbial diversity by surface type. And in the first panel, we see an ordination where each point that you see corresponds to the composition of a particular sample, and the colors are the sample type or the surface type. So what we see is that, and how you read this plot will be in like the closer the points are, the more similar. So what we see here is that different surfaces show different microbial composition. Okay, you see that, for example, escalator handrails that are the black spots tend to be together and in a separate space than other surfaces. For example, the train seats, that would be the green one. So that means that each of the surfaces have their own composition and characteristics. And here and that will be measuring diversity, alpha diversity. And what we see is that platform floors followed by escalator handrails were the most, and the surface with the highest Shannon diversity, while stair handrails and poles were the surfaces with the lowest alpha diversity. So here, what we think is happening is that this is very related to the type of surface, the type of material, and surfaces like poles that are very smooth and from stained steel material, and can it's more difficult that it adheres bacteria instead of platform floors or escalator handrails that are more surfaces with higher porosity. So it allows more bacteria and nutrients and more humidity to stay there and promote growing. So here we have beta and alpha microbial diversity in the next slide among women only and regular wagons. So here we did the comparison, and what we found was that they were not significantly different, both in these two wagon types. And here in the first plot, we have poles and trains in triangles and circles, respectively, and train seats. And for both cases, we didn't find any significance in composition, but in the Shannon index diversity, we didn't find it either. So, and we also explored specifically vaginal associated bacteria, and we tried to identify it, but we didn't find also a difference among these two wagons. So it would probably be because this sample size is very reduced. It would be interesting to do it with a higher sample size. But also, in this train, when it arrives to the end of the line, it changes of direction. So the female or the women only wagons that were before in the front, now it moves to the back of the train. So the actual time that women stayed in that environment, it was around 25 minutes. So maybe it's not enough time to transfer a signature, a female signature, to the environment. Here we have the microbial source. What we did here was trying to infer the source of those microbials in each of the sample that we took. So here we have different bars grouped by surface type, and we have here the proportion of a particular source. And what we see here is that most of the bacteria came from saliva, skin, and soil sources. So that this is the same that we saw in our previous study, and it is a very particular finding in that the Mexico City have that much soil in their surfaces compared with other cities that have already been studied. So here we have microbial colonization after surface cleaning. We have in the first panel poles and train seats. Train seats are the triangles, and they are only there as a reference purpose. So we have that poles that are the circle. They are in different colors, and each of the colors correspond to one point and one time point. So pre-cleaning, we took a sample pre-cleaning, that it will be the blue one, and then we took a sample immediately after cleaning. That is, it doesn't appear here, and it doesn't appear because we had so low, or didn't have that much DNA to amplify it. So that's why we don't have it here. So that means that we really could clean the surface. But then we have in half an hour, samples from 2 hours, 8 hours, and 48 hours. And what we see is that even though we have a separation of groups, significantly separation of groups, those groups and the microbial colonization after cleaning didn't show the pattern of ecological succession in at least 48 hours. It seems that every group, every surface could arrive or could get to a certain point of composition characteristics, but it wouldn't get to the initial characteristics of the microbial composition. And in the panel C, the next one, we have the box plots and with the culture-based analysis, and we have here in CFUs that will be like the number of bacteria or colonies before traveling and before cleaning. And then you have that immediately after cleaning, we have a very low number of bacteria, and then 5, 10, 20 minutes, it will be like a little bit like growing, increasing in number of bacteria. And what we can conclude here is that you only need 20 minutes to reach a similar amount or a similar number of bacteria than pre-cleaning levels. So it's a very rapid process, and that makes us think of how frequently we would have to clean the surfaces for having really a reduced amount of bacteria. So it would be very interesting to perform the same study, but in a more porous, a surface with a higher porosity, where maybe the cleaning would be more useful. So here, now I'm going to present the results with the passenger surface interaction. So we first perform a behavioral description. We calculate the percentage of passengers in contact with different surfaces with their hands. So we first observed around 5,000 people going up and down the escalators and the regular stairs. And we also observed 120 people traveling inside the wagons, inside the train. So here you have the percentage of people, and the colors represent how many, or the percentage of people that touches each of the surfaces. And what we see is that pole, escalator handrails, and the passenger face were the most touched surfaces. You have here the face of a person that is around, that people usually touches. Around 20 and 8% of the people touches in their own face. We also calculate the frequency of hand contact with surfaces every 10 minutes on a train trip. And we saw also that poles and face were the most frequently touched surfaces. So we have that poles were touched around 4 and 5 times every 10 minutes, okay, per person. And that face and also regular personal articles as a whole was touched around 2 and 3 times every 10 minutes. Here, the mucosa, that will be the blue one here, and it includes the mouth, the nose, and mucosa of the eyes, was touched around 0.7 times every 10 minutes. That it's actually a very high frequency, considering that mucosa are the first the area that are in direct contact with our immune system, and it is the most exposed, and it can it occurs the colonization of bacteria on sometimes pathogens. So here we calculate and we study the interchange of bacteria during the subway trip. And what we saw is that in general, we had a mean of 570 bacteria or OTUs. And after the trip, we increased that number to 958. Okay, so we are losing around 400 bacteria, but we are gaining a high amount of bacteria that we are taking it to our house, to the bar, to our jobs. So that's a very interesting finding. We knew it, but having it in numbers make it more interesting. And we also saw that potentially pathogenic bacteria bacteria also increased when you after traveling. So one of the bacteria that we saw that was overrepresented was Arthrobacter cycrolactifilus, that, as you can see before, were not present at all in any of the passengers, and afterwards 7 out of 8 passengers had it. This was discriminant taxa analysis that was performed with Lefse algorithm. And here we also see that increase in diversity, not only in observed diversity, but also in other type of alpha diversity indexes. Here we have an ordination of passenger microbiota before and after traveling. We see that a host microbial signature is preserved. All of these arrows and define the space where different samples are located in these ordination, and each of the colors are each of the passengers, and circles and triangles are the before and after respectively. And what we see is that similar colors that will be similar passengers tend to be together, and that's why is that we're seeing that host microbial signature is preserved. Okay. But we also see that all the triangles tend to go to the right and to move to the same direction. They are a little bit at the right and at down and right of the circle. So they are moving towards the same, having the same microbial composition. And we can see it in the next slide more easily. We have here a heat map where each of the columns represent each of the passengers, and in the next panel, in the same plot, we can see the same passengers, but after traveling. So you have before and after traveling. The color, the blue color, represents the relative abundance of each of the taxa that are each of the rows. Yeah, you can see here. And the orange boxes are the absent taxa. So you can see here that shared bacteria, the number of shared bacteria increases after traveling. So when users, passengers here are not sharing that many bacteria, then after traveling they get it from the train, from the traveling. So this is another sign of, or another type of representation that can be interpreted as they are becoming alike. And they are not just becoming alike among themselves, but also among the surfaces of the subway. So in the first panel, we have all the different surfaces. As it is the ordination that we already saw before, but here is the same one than this, but they are in gray. All the circles are in gray, light gray. But we have here overlapped all the passenger samples, and what we see is that they, in the arrow, is directing from before to after traveling, and they are moving to the middle of the ordination. So yes, to the ordination. So they are also being more similar, or becoming more similar to the composition of the surfaces of the subway. So that's a very nice result. So as a conclusion, We have the Mexico subway is dominated by human skin saliva and soil bacteria, that the floor and escalators have the greatest diversity, and that the tubes, that will be the poles, and seats the least. Poles of wagons are quickly colonized by approximately 20 minutes after cleaning. However, the composition does not appear to recover pre-cleaning characteristics in at least 48 hours. Hand microbiota composition converges among passengers after traveling and becomes more similar to the subway surfaces. However, each passenger preserves his original signature. And after traveling, on the subway, passengers acquire soil and environmental marker bacteria. And as final words, the subway surfaces and passenger microbiota showed the effect of cohabitation with both parties and becoming more similar. The spread of bacteria, the arrival of new ones, and their removal are part of the normal subway dynamics. Therefore, studying them gives us tools to better understand the microbial dynamics of public transportation systems. I wanted also to show you that we developed, with the help of Datametrics Consulting, a web page where general public can explore the results of the microbiome of the subway. So we have, for example, two types of catalogs. Here you can see the friendly catalog that will be for general public, and then it has also the option to download a more specific and more a catalog that can be used for a more deep analysis. Then you have different information in la zona de divulgación. Then you have frequently asked questions. You have a resume of other microbiomes of the world. You have a trivia that will be like a game of questions. And here you have like an example of what you can see in the area of divulgación, where you have here different colors representing the different bacteria, and then that after you touch the poles, you have them. So I would like to end here, and I will acknowledge the Sistema de Transporte Colectivo Metro, to all the students from UNAM, UNAM and UACM also that help performing these studies, taking samples, collecting data, and to the students of el Laboratorio de Genómica Ambiental de la Facultad de Ciencias UNAM, and Ernesto Hecaña Hernández, it's director of the area of culture inside the metro, the subway, and to Datametrics Consulting. This study was financed by Universidad Autónoma Metropolitana, as I told you before, but also by la Secretaría de Educación, Ciencia, Tecnología e Innovación. And we are here, all the members of the lab. So, obrigada. Thank you very much for listening and for the invitation. So I will take any questions.
Oh, thank you very much. I'm gonna clap from here. Thank you very much, Daniela. It was a very, very interesting work, and I'd read a bit about it, but now seeing the whole presentation and you explain it and the website, I thought it was a really cool thing to give back to society, to people interested in it. If you could also, I don't know, if you could share the link, I would like to check out that website a little bit more.
Sure. Sure.
Okay, I think I've got a question here. I'm gonna show it in the screen. I don't know if you see it, so you've probably been getting
yes,
this
I see. Do you think it is possible to extrapolate the result of microbiome location, especially of saliva bacteria, in any way to SARS-CoV-2? Yes. I think we can do it. And one of the things that I would think is that maybe cleaning those surfaces that were mostly dominated by saliva bacteria, that would be the balls, that I don't know why, but it has a lot of saliva. Yes. And so yes, it will give us a idea of how the frequency of cleaning of those surfaces. For example, this type of surfaces, even though they are not collecting that much bacteria, they are, and you can find a lot of saliva bacteria. So maybe they are not almacenando en tanta bacteria, such as maybe probably a more porous surface, such as the escalator handrails that is made of rubber. So the porosity will be like a very, the perfect area to keep accumulating bacteria. So yes, I think that about the cleaning, it can be thought in that way.
Okay, got a question here from Julia. It's a comment. I think it's this comment, and then there is a question.
It was cool to see that the microbial composition commuters from Mexico City's metro is exposed to our, in some sense, healthy composition of human microbiota.
And then
Sure.
she makes this question.
What would you expect for different forms of public transportation? For example, buses. It is very interesting because in the subway we don't have light, so I would wait that in these buses you would have a sterilization process occurring more frequently, just because you are in the presence of the sun. So we don't know what they are there, but in this moment, and we have but since 5 years ago, it has been at this consortia, there is an international consortia, have been collecting data from different transportation systems, and the idea is that you can start analyzing all this data that were collected with the standardized protocols, and you can analyze it as a whole. So comparing also the buses with the subways, with other trains that can be going in the exterior, by the exterior. So yes, I think that it would be very interesting to do this comparison, to know what are the differences. But I would expect some differences.
Daniela, thank you. I think I want to ask you something. So I'm not sure if I got it right or not. But so you were saying, so when you did the time, you follow like colonization times. You kind of mentioned that there were no patterns of ecological succession, although after 28 hours, but in 20 minutes you have kind of the same amount of.
Right. Yes, and. There is ecological succession actually occurring there, but what we expected to happen is that it would get to the same point or the similar composition that in pre-cleaning levels. So we suppose that, okay, so we didn't find that, but the one that you are saying is the with a different approach. We just counted the number of bacteria, yes, that were present. So what we saw is that we didn't know in that for that approach, that's not for that analysis, we didn't know the composition, but what we knew was that at 20 minutes we got the same amount of bacteria that we got pre-cleaning
Okay,
at the
but
pre-cleaning levels.
it's a highly variable community then, like in 20
Right.
minutes you
When we
got a,
see the composition, yeah,
okay,
it is not the same composition. Yes, that is a very good question. Mm-hmm.
okay. So but in this sense, like, so you say, oh, I think there is another question here. Um, I'm gonna ask the question and then I'm gonna show
Yeah.
the question. So when you said, oh, so you have to clean it every 20 minutes, these steel surfaces, right?
Yeah.
But you talked about how porosity surfaces might have like a better work at I don't know, maintaining a more diverse community, and maybe you would least need a less lower times to be cleaning. So why would that be?
In Yes, I think that why that more smooth surfaces would need a higher frequency of cleaning. Not a higher, but in terms of I think that the smooth of the surfaces would tells us how is the velocity of the rechange of the exchange, the rate of exchange. So while you have a very smooth surfaces which each of the passengers touch it, and then the next one will take it out all the bacteria that the one before left. You have other surfaces that are more with a more porosity that will leave bacteria there, and then you have the next passenger that will leave bacteria there, and you have an accumulation of bacteria
Okay.
there. So in that type of surfaces is that I'm thinking that it will be more important to do the cleaning of those surfaces, because you have accumulation instead of those smooth surfaces where the next passenger will rip out or take away the
Whatever
bacteria
was
that
left.
the one before just left. So yeah.
Thank you. There's another question here from Joao.
Yeah. If you conducted this study today when most people are using masks in the subway, would you expect to find less diverse microbiome? Why? By the way, very cool. Thank you. Yes, I would expect a very different microbiome because of that, because we saw that most of the bacteria were from saliva. So when you have this mask where you retain all the saliva, yes, I think that we will have a different composition, maybe, and mostly composed by soil and nice mask
Thank you.
by soil and yes, dust.
Good question, Joao. That's great. So I've got one again, like going back to that porosity thing. So I saw that, for example, I was seeing the other day a program on microbial communities in cheese, so macerated
Okay.
cheese, and they were actually saying that how eventually you could there's people thinking about how you could manage the microbial biota to enhance, like, I don't know, healthy or I say that, like responses to health, like to enhance your immune system through the management of the biota that you're ingesting in cheese. So therefore, you can improve people's um, health system, like immune system. So I don't know if you have seen something about this. I don't know. I don't know much about microbiomes and microbiology. But do you think we could eventually think of like through surfaces, like porosity surfaces, like we could enhance the microbiome of people, so they actually, like, when they go out and they take all these new microbiome, we could eventually be enhancing people's immune system after visiting the subway. Does it make sense,
Yes,
the question?
I think that maybe what you are thinking is more about adaptation. We will get more adapt to our environment. So the bacteria that you usually are in contact with are the ones that are in your environment. So and maybe a person that is not Mexican and usually don't use the subway one day goes to that subway and maybe they are not, and or he is not and used to several type of bacteria, maybe soil bacteria that usually you find it in those surfaces. So maybe you would have a immune reaction. No, I'm not talking about a big immune reaction, but maybe a small immune reaction that you are not aware of, but that your body is then recognizing for the first time or in meeting for the first time. So yes, every time you will be more adapt, or your immune system will know better your environment. Yeah, but yes, I think that it will be a very interesting way to put it, to like going in the subway of Mexico City to get like to get vaccinated, something like that, with the microbiome of the Mexico
Come out
City
with a
subway.
better, you come out of the subway with a better microbiome.
Yeah, right. And yes, one of the things I think it was the first question that they commented, that yes, we are in contact with a lot of bacteria, but that most of them are beneficial or they are neutral, and and we also found that we had an increase of potentially pathogenic bacteria that for people that are healthy, for young people, people that are not immune compromised, it's okay, it doesn't happen not anything. But for maybe vulnerable people, immune suppressed people, or even if you have a wound, an open wound, it would be uh, an important issue, right? But you become vulnerable to this bacteria, this, yes, potentially pathogenic bacteria. Have you heard of any study that investigates the impact for human health in the subway? Yes, there are a lot. I think you are meaning in the subway, I think, right? What we know is that, in like maybe relating it to illnesses, that we have a higher amount of particles, of small particles suspended in the air inside the stations and inside the train in comparison with the exterior. So we are breathing constantly more dust, okay, when we are in traveling the subway. That is at least in the Mexico City subway that doesn't have air conditioning and ventilation. It's mechanical.
So I think we have time for one last question from Andres.
Can you tell us more about pathogens you found like what diseases are expected to find in the subway. We found, when I say pathogenic or potentially pathogenic bacteria, I am talking about maybe bacteria that, including those bacteria that probably 60% of the people have it. So, and in terms of knowing actually what are the pathogenic bacteria, we would have to perform a different study with a different approach. We could detect in general up to the general level of the genus level. Some of them we could and identify to the species level, but to actually go for the pathogen information, we would have to do it at the strain level. And these type of studies are not in that direction. So you would have to choose a particular nutrient, a particular agar, or yes, for isolating a particular pathogens that you know that grows in that media. So yeah, I cannot answer that question. But we found one bacteria in the platform floors that were associated with rats. That was Listeria. We couldn't find the species, but the genus was associated with rats. Mm-hmm.
And is it associated to any disease, or it's just
with disease? Yeah, Listeria. Yeah, with diseases. But we would have to know the species. Mm-hmm.
Yes.
Yeah. And that's very interesting because the subway of Mexico City doesn't have any banks or any seats for waiting in the platforms. Okay. So usually people sit in the floor, and when you have to stand up, you always put the hands over in the floor. So you have their direct contact with your hands, and then your hands goes everywhere. Yeah. So I think that it's not dangerous traveling. We have been doing it always, but we have to be now more aware of where we put our hands, as we already know. And if we are not aware of that, well, you put some alcohol, and now you can be more relaxed.
Now I feel more relaxed. So, well, I don't know if there's that for another question, but if not, thank you, Daniela, once again very much for having.
Thank you, Adrian.
This work. It's really, really, really cool. I was just wondering, are there many studies in subways?
Around with these techniques, around 12 studies have been performed in different cities, but there are a lot more that they have been developing very fast.
Cool. Well, thank you very much, and obrigado a todo mundo que assistiu. Quem quiser, semana que vem tem mais.
Bye. Gracias.